Hello everyone, welcome to masterspharma. Today we'll discuss about some pharmacognosy MCQs. Here we go. The first question. Alkaloids show strong physiological activity mainly because they contain options: aromatic rings, hydroxyl groups, nitrogen atoms, carboxyl groups. The correct option is option C, nitrogen atoms. Alkaloids show strong physiological activity mainly because their nitrogen atoms can accept protons and form cations, allowing them to interact strongly with biological receptors and enzymes. Now we see the other options. Option A, aromatic rings, common in many molecules but not responsible for unique strong physiological activity of alkaloids. Option B, hydroxyl groups, affect solubility and reactivity but don't define alkaloids activity. Option D, carboxyl groups, rare in alkaloids, generally make molecules acidic, so not related to their biological effects. So the correct option is option C, nitrogen atoms. The next question. Morphine is an example of dash. Options: synthetic drug, semi-synthetic drug, natural drug and biotechnological drug. The correct option is option C, natural drug. Morphine is a natural drug because it is directly obtained from opium poppy known as Papaver somniferum without any chemical modifications. It is an original plant alkaloids found in nature. Now we see the other options. Option A, synthetic drug made entirely by a chemical synthesis. It is not true for morphine. Option B, semi-synthetic drug. It is chemically modified natural components. Morphine is unmodified. Option D, biotechnological drugs produced using biotechnology or living system. It is also not applicable. So the correct option is option C, natural drug. The next question. Mixing exhausted ginger with genuine ginger is a type of dash. Options: refinement, substitution, sophistication and dilution. Here the correct option is option B, substitution. Mixing exhausted ginger with genuine is considered substitution because the exhausted ginger is replacing part of the genuine drug with a cheaper, inactive, inferior version of the same material. So the correct option is substitution. Now we see the other options. Refinement, improving purity, it is also not applicable. Option C, sophistication, adding substances to mimic good quality. So it is also not correct. Option D, dilution, adding inert materials. Exhausted ginger is not an inert. So the correct option is option B. The next question. Which method is best for evaluating the volatile oils? Options: TLC, thin layer chromatography, distillation, maceration and percolation. The correct option is option B, distillation. Distillation, especially a steam distillation, is the best method for evaluating volatile oils because it separates and quantifies the volatile components without decomposing them. It is the standard pharmacopoeial method for determining the volatile content. Now if we see the other options. TLC, thin layer chromatography, used for identification, not for quantifying volatile oils. Option C, maceration, for extracting volatile oil constituents. Option D, percolation, it is used for preparing tinctures and extracts, not for the volatile oils. So the correct option is option B, distillation. The next question. Which is an example of marine drug source? Options: cod liver oil, castor oil, olive oil and clove oil. The correct option is option A, cod liver oil. Cod liver oil comes from the liver of cod fish, making it an marine. It is an animal drug source as it is directly obtained from the organisms living in the sea. Now we see the other options. Option B, castor oil. It is from castor seeds. It is also a plant, not from the marine. Option C, olive oil. It is from also a olives. It is also a plant, not from the marine. Option D, it is from clove buds. It is also a plant. It is also not from the marine. So the correct option is option A, cod liver oil. The next question. The primary purpose of secondary metabolites in plant is options: human medicinal use, defense and survival, seed production and soil nutrition. The correct option is option B, defense and survival. Defense and survival is a secondary metabolites like alkaloids, tannins, flavonoids and terpenoids are produced by plants mainly for defense against herbivores, microbes and environmental stress and to aid survival through pollinator attractions, UV protections and competitions. So the correct option is option B. Now we see the other options. Human medicinal use. This is only a benefit to humans, not the plant purpose. Option C, seed productions. It is done by primary metabolites like proteins, carbs and fat. Now we see the other option. Soil nutrition, option D. It not the function of secondary metabolites. So the correct option is option B, defense and survival. The next question. Why does atropine cause dilation of pupils? Options. It stimulates adrenergic receptor of the iris. It blocks muscarinic receptors in the sphincter pupillae. Option C. It activates nicotinic receptors in the ciliary muscles. Option D. It increases aqueous humor formation. Now we see the correct answer. Correct answer is option B. It blocks muscarinic receptors in the sphincter pupillae. Atropine is a muscarinic antagonist. By blocking M3 receptors in the sphincter pupillae, it prevents constriction. So the opposing muscle becomes unopposed and pupil dilates. So the correct option is B. Now we see the other options. Option A. It stimulates adrenergic receptor of the iris. Atropine does not stimulate adrenergic receptor. Option C. It activates nicotinic receptor in the ciliary muscles. Atropine does not act on nicotinic receptors. Option D. It increases aqueous humor formation, not related to pupil. So it is also a not correct answer. Now the correct answer is option B. The next question. Cardiac glycosides acts by inhibiting. Options: calcium channels, sodium potassium ATPase, potassium channels and G proteins. The correct option is option B, sodium potassium ATPase. Cardiac glycosides like digoxin inhibit sodium and potassium ATPase pumps in cardiac cells. This increases intracellular sodium, which reduces sodium and calcium exchanger activity. It leads to causing more calcium to stay inside the cell, leading to strong heart contraction. Now we see the other options. Calcium channels. It not directly blocked by cardiac glycosides. Option C, potassium channels, not the primary target. Option D, G protein. Mechanism is not a G protein mediated. So the correct option is option B. The next question. Digitalis toxicity occurs due to the drugs has dash. Options: weak potency, high safety margin, narrow therapeutic index, no effects at high doses. The correct option is option C, narrow therapeutic index. Digitalics. Example, digoxin has very small difference between the therapeutic dose and the toxic dose. Because of this narrow therapeutic index, even a slight increases in blood vessels can lead to severe toxicity. So the correct option is narrow therapeutic index. Now we see the other options. Option A, weak potency. Digitalis is actually a highly potent. So it is not correct. Option B, high safety margin. No, digitalis has low safety margin. Option D, no effects at high doses, but it has a dangerous effect at high doses. So the correct option is option C. The next question. Morphine reduces pain mainly by acting on dash. Options: dopamine receptors, mu opioid receptors, H1 receptors and GABA receptors. The correct option is option B, mu opioid receptors. Morphine relieves pain primarily by activating mu opioid receptors in the brain and spinal cord. This inhibits the pain transmission, increases the pain tolerance and produces analgesia. Now we see the other options. Option A, dopamine receptors involved in reward, not the primary analgesia. Option C, H1 receptors histamine receptors unrelated to pain relief. So it is also a not correct answer. Option D, GABA receptors. Inhibitors neurotransmission but not morphine's main targets. So it is also not correct. So correct option is mu-opioid receptors. Yeah, that's for today. Thank you for watching. For more such content, please follow us on Instagram and subscribe to our YouTube channel Masters Pharma.